Hello, welcome to AP Bio Review Video 12. This one will be a short video. It's an application of some cell communication related things in the immune system. And so what we have here is, it'll be a little bit dark because this is so bright, but once we're done watching this video, I'll turn it off. What we have right here is a zebrafish, and they basically go in and they take a zebrafish and they create a wound in the side right here, and what we're looking for is an immune response. So essentially, if I damage my skin, I don't know why it's taking me so long to think of this. If I got a cut right here or something and damage my skin, then nearby white blood cells actually respond and they go to the site of the puncture wound. And so what we're going to do here is we're going to first see that happen in the video, and then we're going to explain how it happens through cell communication. So here's the wound that's introduced. If I remember right, they label it. And what we'll be looking for is, coming from this blood vessel right here, we're going to look for white blood cells, immune system cells that respond. By the way, the zebrafish was anesthetized in the video. So there you go. I guess they have the arrows. We're seeing white blood cells leaking out, and they're all going to that particular site of infection. They're going to zoom out, and they're zooming out just so you realize that the white blood cells are only going to that site. They're not just randomly leaving the white blood cells everywhere. They are specifically going to the site of infection. Almost like somebody's yelling and they are responding to the call. So what we want to do is figure out the cause for that. Thanks, Michael and Paul. So if we look over here, and the humming sound of the projector will go off in just a moment. If we look over here, here's the surface of my zebrafish, and there's a cut. So there you go, there's a cut. This is a blood vessel. Now if this is a capillary-like blood vessel, capillaries are only one cell layer thick. And they're also made of the thinnest cells that we've got. In fact, the cells are so thin that they can be thinner than the nucleus of the cell itself. You might wonder how that's possible, but essentially they look sort of like a fried egg. Like that. So right there, the nucleus is that thick, and the cell is actually only that thick right there. So this is called a simple squamous or a simple squamous cell. Let's unhook this. There we go. So over here, these are really thin cells, and they're only one cell layer thick. They're not stratified. I don't have 30 cell layers there. So that's my capillary. So blood's flying through here, but also white blood cells are cruising through here, immune system cells. We're not concerned with all the different types. When we do an immune unit, we'll look at the difference between a phagocyte and a mast cell and all the phils, neutrophils, basophils, eosinophils, possibly. But for now, we're just going to kind of assume that white blood cells are part of our immune system, and they're just helping me fight infection. So what will happen is maybe some bacteria get in here. So bacteria move in. These guys are some kind of rod-shaped bacteria, maybe E. coli or something, and they enter in through the cut. Okay, so now my cells have to respond. So some white blood cells might just happen to bump into them. And what will occur is those white blood cells then want to send out a chemical signal of like, hey buddy, hey everybody, we're being invaded. So think of it this way. If I'm running security and all of a sudden I turn the corner and I see that there's a bunch of bacteria, there's probably not just one or two, there are probably thousands or millions of bacteria. So I don't want to try to fight them myself. Certainly I could take down a few dozen, but I probably can't take down thousands of bacteria. So I want to call for help. And what we'll do in order to make that happen is we have what are called chemokines. Chemo, it's a chemical, and kine, we see this a lot, kinesis, kinetic, movement. So it's a chemical that recruits cells and has them kind of follow the chemical scent. So think of it as like a diffusion. I'm sending out a call, And if you think of diffusion, these guys can sort of follow the scent as they go from lower to higher concentration. Back when we used to have choir cookies before we couldn't do that, if I was blindfolded, my room is upstairs, the choir cookies were downstairs. If I was blindfolded, I could kind of follow the scent. If I start to make a wrong turn, the scent gets lighter and lighter, and then I turn around, and as the scent gets stronger and stronger and stronger, I find the source. So that's how chemokines work. Well, these guys are now going to go to that area of the infection, if they can. The problem is that they're flying by in the bloodstream with capillaries. So now what we do is we release histamines. I'll make histamines green. Histamine. So whenever cells, some of these immune cells that are fighting, will release histamines, that is what makes my blood cells leaky. That's what makes my capillaries leaky. So the way that happens is this, and this, and this. Let's say that they each contract by like 2 or 3%. So right now, these 4 cells that make up the wall of the capillary are all shoulder to shoulder, so to speak. But what if that guy contracts by just a little bit, and that guy contracts by just a little bit, and that guy, and that guy? Now we just sort of opened up a gap, right? If I've got these capillaries right here, like this, and it's a nice fit, but if all of a sudden that green pin and the blue pin and the red pin each shrink by 5%, now there's like a gap in between. And so now these guys can actually leave and go towards the infection. If you've had an anatomy class, you might have seen like diapedesis. That's like squeezing through the capillaries. So histamine basically is making my capillaries leaky. So I'll say leaky caps. And chemokines are basically saying, over here, help. I don't know if my acting paid off there. I've never had an acting lesson. So over here, help. Histamines make the capillaries leaky. In addition, we have prostaglandins. Oops. I'll make those a different color. Prostaglandins. Prostaglandin. It ends in IN, so it's probably a protein gland. And then we probably discovered it in the prostate gland, I'm assuming. And so prostaglandins, they cause the capillaries to dilate. So now instead of being this big, they'll be this big. They're increasing blood flow. So now hopefully more white blood cells can get there. Because of histamine, the white blood cells can leak out. Once they leak out, because of chemokines, they know where to go to fight. And so now they're basically backup that are going to fight. Prostaglandins can also provide some of the like itchy feeling that you feel, like if you get a mosquito bite, the itchiness. Part of that is from prostaglandins. And these are not the only chemicals released. These are just some of them. Another one, pyrogens. Now pyrogens are kind of a general term. Sometimes the bacteria themselves have proteins on their surfaces that act as pyrogens. But my white blood cells, my immune system cells, can also release pyrogens. Pyro is fire. So pyrogens are the cause of increasing, that's supposed to be an up arrow, increasing fever. What's going to happen is these pyrogens get dumped into the bloodstream, so they're released also. So now these pyrogens, I'll just put pyro, these pyrogens eventually make it up to my brain, and various regions of my brain kind of had different tasks. Some tasks are spread everywhere. But one of those regions is the hypothalamus. You wouldn't have to know that for the AP Bio test. It's hypo, it's below the thalamus. And the hypothalamus does a lot of like kind of basic homeostasis type of activities. One of them is maintaining our body temperature. And in my brain, in the HT, the hypothalamus, I'll receive those pyrogens. And when I start receiving pyrogens, my brain will start like slowly upping my body temperature. So let's say that my typical body temperature, we always say 98.6, but not everybody's exactly that. Let's say that mine's 98. Well, when pyrogens start hitting my hypothalamus, then now my body temperature goes up to 98.1. More pyrogens, 98.2. More pyrogens, 98.3, 4, 5, 99, 100, 101, 102. So the more pyrogens there are, the higher my temperature goes. Well, why would we release more pyrogens? The more immune cells that are involved, the more pyrogens we're going to release. So pretend for a second that this guy releases pyrogens because he's interacting with bacteria. And then let's say that these guys, once they start interacting with bacteria, I'm simplifying a little bit, then they release pyrogens. Well, a small infection means we'll have a small response. There'll be a small number of cells. And so we'll release a small amount of pyrogens, so I get a mild fever. If I have a major infection, there are more cells involved, so they're releasing more pyrogens, so my temperature goes up. So that's one of the reasons why, let's say you get a fever, are you supposed to go to school tomorrow or not? Well, how high was your fever? And so different schools will have different rules, but it's like, hey, if you've had a fever of 100 or higher, then you need to be below 100 for 24 hours before you return to school or something like that. Why does the fever matter? A fever is an indicator of how bad the infection is. Mild infection, mild fever, 99.5, but fever of 102, that's probably a major infection. Don't come to school, you're going to infect us all. From here, this also kind of explains allergic reactions. So me, as I've said before in class, ironically, I'm allergic to cottonwood tree pollen. And so whenever my body encounters pollen, my immune system thinks that it's a foreign object. It is a foreign object, but it's pollen. Pollen comes from plants, so it does have cellular components. It's got, glycoproteins and stuff like that. My immune system checks those glycoproteins, doesn't recognize what they are, and it starts doing this. So let's say that I'm allergic to peanut butter. I get a cut, I rub peanut butter in it. Why? Because I'm a freaking idiot. Or I eat it, and now my tongue starts to swell. So a local reaction right there, quick aside, that's what we do for allergy testing. For allergy testing, they'll basically get little bitty serums of all the things that we're typically allergic to, whether it's ragweed or peanut or something like that, and they basically just sort of scratch it. They'll make kind of a grid on your back, or at least this is how they used to do it, and they just sort of scratch in a little bit of that. So I have a little needle, I dip it in ragweed pollen concentrate, and then just kind of poke your skin and see if there's a reaction. Well, if there's a reaction, it'll get all swollen and red, right? If I get an infection, if this is a cut, and this is a cut, and this one's infected and this one's not, I'll know this one's infected because it'll be all swollen and red. Well, the reason for that is this. Histamine is causing leaky capillaries. So white blood cells leave, but can red blood cells also squeeze through there? Yeah. So now that area starts to kind of swell up with fluid. Excuse me. And that area swells up with fluid, and if red blood cells are leaking, it looks red. And so that's why that red puffy area is red and puffy whenever I have an infection. Back to allergies. If my body thinks that ragweed pollen or cottonwood tree pollen is bad, then whenever it bumps into it, it's going to do these things. It's going to release histamine. And if this is happening up here, that's why my sinuses just feel like swollen and full and everything else. So what I can do is I can go to the store and buy some antihistamines. And it'll do the opposite. It'll basically reduce swelling by closing the capillaries back up. Now, if I had a real infection, something that could harm me, a bacteria that's actually a pathogen or something, then swelling this is my body responding properly. With allergies, ragweed pollen or cottonwood tree pollen can't actually harm me, so I just want to treat the symptoms. But the thing is like, I breathe in and my body's like, we're being invaded, make histamine, make prostaglandins and all that. And then I take another breath of, the air with pollen in it, and they're like, we're being invaded again. And then I take another breath, we're being invaded again. And so it just gets worse and worse and worse. my body will start to make my eyes kind of watery because they're like, you know, we're going to get invaded. It's going to get in his eyes. Wash it out, wash it out. My body will start making a bunch of snot because it's like, all right, we don't want it to get into his lungs. He keeps breathing it in, you know, captured in the snot, captured in the snot. And so you're snotty all the time. Vaccines. Let's say I get a vaccine. Hopefully we'll have a vaccine for SARS-CoV-2 sooner rather than later. Sooner might be a year, later might be a year and a half, or never. Hopefully we get one in a year or so. And whenever I get that vaccine, sometimes people will say after their vaccine, a day or two later, after a flu vaccine or something, they feel just a little bit under the weather. Well, your body is still reacting. If we give you some destroyed flu virus, then your body is still going to do all of this stuff. We're giving you a small amount though, enough to where you have an immune response, but small enough to where it shouldn't like give you a fever of 103 or something. So you have a mild response, your body does this. So sometimes people say, oh, I got a vaccine, and so now I feel like I have the flu, but it was just kind of mild. No, that's your body reacting. Then the next time that you're exposed to the real flu, let's say I get a vaccine for flu of 2025, and then five weeks later, I'm exposed to the real flu, I've got a built-up immune response. I have antibodies already built up for fighting the real flu. All right, our next video, we'll get into DNA structure and DNA replication. So thanks for coming out.